So Fred, let's talk about CAR T cells. This is a novel cellular therapy. It seems like there's been almost instantaneous development. In reality, these therapies have been worked on for decades now. There's been some major changes in how these therapies are designed and resulting in an increased efficacy. Can you talk to us about that?
Absolutely. So CAR T cells, or chimeric antigen receptor T cells, essentially refers to the insertion of a gene that would express a protein within otherwise polyclonal T cells. And the expression of that gene or that protein has an extracellular domain that is derived from an antibody, so it can bind to whatever it's designed to bind against. It has a hinge or transcellular domain, and then it has an intracellular domain that provides a signaling mechanism to activate that T cell. The first generation CARs were described all the way back in the 80s by Zelig Eshar, and so it's been a long time coming, as you mentioned. The second generation CARs, what the second generation CARs do is they don't just include the CD3 zeta signaling domain, which activates T cells, they also include the co-stimulatory domain, the second signal to activate T cells. There's several different co-stimulatory domains that are in use today. There's a CD28 co-stimulatory domain and a 41BB co-stimulatory domain. And what's happened is that individual investigators and groups at different labs around the country and the world have come to find that CD19, which is on the surface of B cells and B-cell malignancies, serves as really a model and a perfect target for these chimeric antigen receptor T cells. So anti-CD19 CAR T cells were developed at these individual institutions and were shown to have great success against B-cell malignancies like B-cell ALL, diffuse large B-cell lymphoma. And what's happened since that time is that drug companies have come and licensed those constructs, those genes that express those CARs. And so now we have numerous drug companies developing these, with really three quite advanced towards getting approval, or at least in trials seeking to get approval, FDA approval of these chimeric antigen receptors T cells targeting CD19. The therapy, what it involves for a patient is that they undergo an apheresis. They have to have their T cells collected. So not unlike what we do for an autologous hematopoietic stem cell transplant, the patient goes on a apheresis machine, they have their cells collected, and then that gene is put into those otherwise polyclonal T cells to redirect them against, in this case, CD19. And then the cells are infused after lymphodepleting chemotherapy. So the lymphodepleting chemotherapy drives down the existing lymphocytes that are within the body, creates space for those transferred T cells, and then once they're infused back in, they proliferate. Generally, there's a spike in the number of those CAR T cells within the patient, and then they act as T cells do with these CAR constructs to attack the CD19 bearing tumor. So it's pretty exciting times. There's been amazing responses.
What's very impressive, clinically very powerful, we had work with Kite and Jim Kokendorfer and then Steve Rosenberg team at the NCI for 7-8 years now, and we had some patients at the beginning that had really extensive disease, refractory large cell lymphoma, who basically were a patient that would be candidate to go to hospice, unfortunately, and who had one injection and remained in remission for several years. There's one patient particularly that was treated on the NCI who had a primary intestinal large cell lymphoma relapse with extrathoracic disease, liver mets, 10 prior therapy, young woman, 35 years old, CAR T cells, 4 years of remission. So this is really something that is very critical.